One area that is emerging at the Salk Institute is our combined interest in aging and how we age as organisms. What's exciting about it for me is that we have between 10 and 15 existing faculty members whose programs are influenced or directed towards understanding how we age as individuals. Each one of these faculty members approaches the problem from a different perspective. And this allows us to work cooperatively together on the different systems that we are working on in such a way that we can unravel the small components and then put them back together to get a better understanding of how we age, why we age, and how this affects our cognitive functioning.
I'm excited as part of the initiative to work across disciplines with my colleagues in cancer biology or immunology to really bring them into neuroscience so we can make big discoveries.
Aging research at the Salk Institute has a long and successful tradition, and it started with establishment of the Glenn Center for Research on Aging more than a decade ago, and it has since continued through support by the Glenn Foundation and also through support by the American Heart Association and Allen Initiative that are both interested in furthering aging research at the Salk Institute.
Aging by far is the biggest risk factor for many common human diseases, heart disease, diabetes, obesity, cancer, Alzheimer's, and other neurodegenerative diseases. But what we don't understand is how aging is the biggest risk factor for each of those individual diseases. So I think a big push in aging research is to try to understand how different cells, tissues, and organs age in order to find the specific pathways in a person that has one or other of those particular diseases.
My lab is interested in understanding how individuals, their organs, and even individual cells within these organs age over the course of a lifetime. The chronological age, so the number that is on your birthday cake, is actually not very informative and doesn't tell us much about how you age. So there could be two 80-year-olds, one just qualifying for a Boston Marathon and the other one spending his or her days in assisted living facilities. And what we'd like to do is to be able to compare now individuals or organs and cells that either have a younger biological age than their chronological age or have an older biological age. And by comparing those two, we hope to identify underlying mechanisms that allow some people to maintain health throughout life, while others are not able to do that.
My lab works on telomeres and the DNA damage response, and we are optimistic that through our approach, by bringing different disciplines together and through the incorporation of computational approaches, that we will be in a unique situation to understand aging and to alleviate age-associated disease. Telomeres represent an intrinsic clock in our cells that only allow them to double a certain number of times, and thereby they prevent unlimited growth and immortality and, of course, cancer development.
My lab's particularly interested in the possibility that we'll be able to identify the factors that dictate how fast the ends of chromosomes erode, and potentially identify how efficiently those factors act in different individuals in a human population. And if so, the big pie in the sky goal is that maybe we could tweak the eroding process very early in life, where we could influence in the first decade of life, for example, how fast an individual's telomeres are eroding so that late in their life they'll live well into their 90s without any of the pathophysiology of aging as the rest of us know it.
My research is actually directed at mitochondria, which are organelles inside most of your cells that actually convert the air that we breathe and the food that we eat into chemical energy called ATP. What my work has shown in that regard is that Mitochondria contribute to aging in two different ways, both positively and negatively.
So the work in my lab, we're trying to understand the brain and how it is that your brain changes with age, either in normal aging or in diseases like Alzheimer's. So you're probably familiar with cells in the brain called neurons, but what we work on are the other cells called glia that actually surround the neurons. And the reason we're excited about these cells is work we recently did showed that perhaps in normal aging and also neurodegeneration, these are the cells that change. And actually these changes in glial cells could be contributing to cognitive decline and the progression of all these different diseases. So as part of these bigger initiatives at Salk, what we're really excited to do is to work with the other groups and ask, well, can we target these glial cells or the changes that happen in aging and see if we can slow down the progression of cognitive decline or even prevent it from happening.
We're right on the cusp of solving some really fundamental problems that I think are going to change the course of how we age.